Place of origin. The presence of mitotic figures near the lumen of the cerebral cavity of the embryonic human cerebrum, and their paucity and/or absence in the cortical plate itself, led to the hypothesis that cortical neurons are produced in the germinal matrix situated at the ventricular surface. Hiss, 1904. An idea that was substantiated by the labeling of dividing cells in mice, and Gavin and Sidman, 1961, monkeys, Rakesh, 1974, and humans, Rakesh and Sidman, 1968. In the last decade, studies in rodents have established two sources of origin of cortical neurons. The germinal regions of the dorsal telencephalon give rise to cortical projection neurons that migrate radially to their final position in the cortex, while the ganglionic eminences in the ventral telencephalon give rise to virtually all GABAergic cortical interneurons, which migrate tangentially via the marginal and intermediate zones to the cortex. This model has been observed in rodents and carnivores, for example ferrets, but also in avians such as chickens, and in a similar population of tangentially migrating neurons has been observed in the human using classical histological material. Rakesh, 1975. However, in what appears to be significant species-specific difference, neocortical inhibitory interneurons in primates, including human, originate not only in the ganglionic eminences in the ventral telencephalon, but also in the ventricular zone and in the enormously enlarged subventricular zone of the dorsal telencephalon. In quantitative data from Letnick and colleagues 2002 indicated that about 2/3 of the cortical GABAergic interneurons might have a dorsal telencephalic origin in humans. Proliferative cells in the ventricular zone are organized in a pseudostratified epithelium in which precursor cells divide asynchronously. Their nuclei move away from the ventricular surface to replicate their DNA and then move back to the surface to undergo another mitotic cycle. Early silver impregnation methods revealed a distinct population of elongated non-neuronal cells in the fetal human brain, which were initially called epithelial cells or fetal glia. Later, electron microscopic and immunohistochemical studies using glial acidic fibrillary protein GFAP in human and non-human primates confirmed their glial nature and justified the use of the term radial glial cells, RGC. The distal end feet of the radial glial cells form the peel surface of the fetal cerebrum. A variety of antigens such as vimentin, brain lipid binding protein BLBP, the astrocyte-specific glutamate transporters, GLAST, and RC1, RC2 have been used to characterize their glial nature. Radial glia is particularly prominent Radial glia is particularly prominent in the embryonic primate cerebrum, where a subset of GFAP-positive cells transiently stops dividing. However, it has become now clear that RGC can generate both neuronal progenitors and neurons. The dividing radial glial cells generate successive neuronal clones that migrate along the elongated parental process using the cortical plate or populate the subventricular zone, SVZ, where they divide again before entering a postmitotic state and migrate to the overlying cortex. In addition, they generate intermediate progenitors that continue to divide. The subventricular zone was previously thought to generate mainly glia. However, it is now well established that this zone is multipotential and also generates projection neurons and interneurons, as well as different types of glial cells in all species studied, although the proportion is different between species. Thus, in rodents and primates, early divergence of basic cell types has been revealed using the retroviral gene transfer method, which enables the study of cell lineages in the developmental mammalian telencephalon. It appears that in the primate, the radioglial mother cell gives rise to a daughter cell which either directly or as a dedicated neuronal progenitor goes through several rounds of division to produce bipolar migrating neurons that will migrate up the radial process of the mother cell. Current state-of-the-art methods, including in utero electroporation and conditional mouse genetics, which Both allow for rapid and powerful gain and loss of function. Studies are beginning to unravel the complex molecular interplay between these cell types in the developing cortex. A complete compendium of newly identified signaling pathways is beyond the scope of this chapter, but a few findings deserve mention. For example, it has been demonstrated that the protein NUMB, N-U-M-B, is a crucial player in maintaining the adhesiveness of radioglia in the ventricular zone, VZ, preventing premature detachment and subsequent astrogliogenesis. In contrast, Notch functions cell autonomously to maintain the radioglial cell plate, while the proneural genes antagonize Notch signaling to promote neuronal differentiation and subsequent migration. More precisely, in an example of the exquisite balance of structure and function, it has been shown that the daughter neuronal cell stimulates Notch signaling in the radioglial mother cell to maintain the neurogenic ventricular zone niche and migratory scaffold.